Hello, my name is Romain Lavaud. I'm an assistant professor at Louisiana State University Agricultural Center in Baton Rouge, Louisiana. And in this lecture, I will talk to you about trajectory reconstruction in dynamic energy budget. I will start with an introduction, explaining what is the interest for such trajectory reconstruction. And then I will give you a series of examples about different variables that we can reconstruct. And I will focus on two examples, especially one about the reconstruction of food intake and growth from fish otolith, and the other one about the reconstruction of physiological rates from scallop shells. But first, what do we use reconstructions for? In science, there's a variety of applications which mostly use features in images, encoded data, or a signal that integrates the impact of some variables on an observed variables in order to produce a result that is can be considered a reconstruction. For example, in botany, we can use satellite imagery in canopy reflectance models to reconstruct some morphological characteristic of forests. Or in artificial intelligence, deep learning in biomedicine is a field that is in development and that is based on the concept of reconstructing deeper structures in the body. Another field where it's used is archaeology, where we're trying to reconstruct, recover more so biological and civilization structures. And of course, maybe the most obvious one for our field of research is in relation with climate change, where in which studies we will use records of past environments in what we call dendrochronology and the sclerochronology. So using wood structure or calcified structure as records of past environment, and in order to reconstruct the past climate. And so in DEB research, trajectory reconstruction is the use of a signal that we have, and using that signal to reconstruct some variables that impacted the physiology of the organism and that resulted in the signal we can observe. And the most common question in DEB studies is related to finding which environmental condition resulted in the observed trajectory. And the term trajectory here can refer to many different things. It can be a growth trajectory in length or in weight. That's the most often used. We will see that there are also other types of trajectories that we can use. And in the case of DEB modeling, because we make use of a mechanistic model, it often means that we will invert the model and sometimes rewrite equations so that we can isolate a term which was a variable or a parameter in this equation to then estimate and reconstruct this variable or this quantity using information that we observed. The applications for such studies are very diverse. We will see examples that deal with trophic ecology, so mostly what food do the organism feed on. It has also some applications in species conservation, in climate change, of course, in fisheries management. That would be the example with the otolith. And it's also, these inversions, these reconstruction can also be a way to check that the mechanistic theory that we use, depth theory, is accurate and correct. Depth theory, as any theory, is a representation of what we observe in the real world. the real world, we measure data on organisms. We also measure environmental conditions, and we're trying to relate these two together. In the fantasy world of the theory, we use these environmental data as forcing variables, and we are attempting to construct observables from equations and parameters that we will be comparing to the data that we observe in the real world. The first step in doing so is to estimate the parameter. A lot of the content of this course is dedicated to the estimation of these primary parameter values. And that's because it's a very important part of this process. But it's a complex process that involves the definition of state variables, as well as their initial values, and the use of ordinary differential equations in the case of depth theory to compute the change of state variables based on the forcing variables and on the primary parameter values. But from there, because we are in this fantasy world in which we represent things that don't necessarily not exist, but are difficult to measure, we need auxiliary equation and auxiliary parameters to transform them into quantities that are considered observables and that we can compare to organism data. And in reconstructing techniques and approach, what we are trying to do is to invert the cycle, and starting from organism data that we observed in the field, in the real world or in an experiment, and move back along the chain of the sequence of events that are required to perform a trajectory construction. So in reconstruction, we go from this organism data and invert the model, or use techniques that allow us to retrieve the original environmental data that were responsible or impacted the metabolism of the organism and resulted in the observations that we now use. So this is what a reconstruction is attempting to do. So now let's move on to some examples of reconstruction in depth. There are many different examples that exist. I will focus on some of them, but give you the references to the other ones if you're interested in that type of reconstruction. But I will focus mostly on the food intake from growth data in two examples for mussels and penguins. And then we'll move on rapidly to another type of reconstruction for food intake and growth from otolith data in anchovy. And then we'll look at another approach that allows you to reconstruct reserve dynamics, reserve mobilization, somatic maintenance from shell growth data, scallop shell growth data. So first, about the reconstruction of food intake from growth data. So you have here a representation, a simplified representation of the DEB model and the input variables that it requires to predict, for example, individual growth. So food ingestion in DEB model and assimilation is usually well explained by a simple functional response, as you see in the graph there. This is actually a graph for Daphnia, but in our example here, we will talk about the blue mussel. And I just give you here two links for very cool videos on YouTube about bivalve filtration and how they clear the water of particle, if you are curious about that. But this concept of functional response is pretty standard in depth theory, at least in the standard DEB model. And this is what we are trying to reconstruct in this case, the scaled functional response. But first, we need to make an assumption, which is that the change in food density is slow through time. So we need to assume that F doesn't vary too much. And if that's the case, we can then assume that little e, which is the scaled reserve density, will converge to the value of F. So that's an assumption that is required in this problem, because we can then rewrite the growth equation and simplify it. And from that simplification, we can get an equation for the functional response. We can isolate this term. And you see now in red in this formula, the two quantities that we need to get to estimate to be able to reconstruct the functional response. And these are the scale length, little l, and its variation through time. And to retrieve these quantities, an easy process is to fit a cubic spline function through the trajectory length data that we have from the environment, from the real world, in order to get these two quantities, L and its variation over time. So cubic spline functions are often used in interpolation, and they rely on the definition of knots. And the more knots you have, basically, the more precise the interpolation will be. And this is the same for our reconstruction. The more knots in time you have, the more precise the reconstruction will be. And then, this spline function, there is a function in the DepTool package that is available and that you can use pretty easily. And so for our example of the blue mussel, you have here everything on the same graph. The growth trajectories are the green to blue dots. Through which we fit a spline function in each of the trajectories. And we also fit a spline function, through the temperature trajectory in red. So the smooth curve is the fitted curve. And from this temperature and data and the growth trajectory, we can reconstruct the scaled functional response in purple here on the graph. And so this is a method that can be used when availability of data about food availability, food concentration, are not easily obtained. And it's also a way to compare the functional response to different proxies of food concentration in the environment. And but in some cases, you cannot make the first assumption that we made about the change in food concentration in the environment. And if food changes too much, then the scaled reserve density cannot be assumed to be equal to F. And in that case, we must also reconstruct the reserve density, capital E in a square bracket, which is the ratio of reserve over structure. And this complexifies a little bit our problem, because we have then to not only take care of structure state variable, but also the reserve, and in this case, in particular, the reserve density. And so, as I said, it complexifies a little bit the problem, because now we also have to estimate the scaled reserve density. First, we need to fit a spline function through the weight data trajectory and retrieve weight and its variation over time. And then we can get little e through time through a numerical integration of the following equation. And in the example that we have here, we compare two species of penguin, the Adelie penguin and the emperor penguin. On the left, the Adelie penguin has a rather constant increase in weight from birth. That's because food is rather constant and the parents can feed the chicks at a constant level. The small drop that you see at the end corresponds to the weaning period. And in the case of the emperor penguin, the situation is quite different because the breeding cycle lasts much longer, between 14 and 17 months. And during this time, food fluctuates in the ocean and the parents are not able to provide a continuous supply of food to the chicks. And that reflects in their growth curve. And this is interesting to look back at this functional and this reconstructed functional response, because it can then help understand the fluctuation of food in the ocean and the fluctuation of these prey stocks, this fish population that the emperor penguin feeds on. And this is particularly interesting for species for which it's difficult to obtain data, such as these penguins living in a very remote area. And there are other types of reconstruction that can be carried out. For example, reconstructing the body temperature from growth data. This is here an example on bird species. And this is relevant for these birds because the body temperature can influence at the beginning, especially the value of the parameters during a parameter estimation. So if you use these growth trajectories in a parameter estimation, it's good to know about the body temperature. However, it's not a quantity that is often measured, and this procedure can help you get that. Another example is the reconstruction of food intake from reproduction data. And we have here an example for Daphnia, the water flea, in which we use the reserve density of the eggs to try to recover the scallop functional response. Note here that this is also a way to check one concept of the theory, the maternal effect, which states that the reserve density of the mother is then found at the same value in the eggs. And so inverting this model here allows to check this principle. All right, now let's dive a little bit more into another example, which is the food intake and growth from otolith data in the anchovy. So now let's move on to another example of food intake reconstruction, but also growth reconstruction from otolith data. This is based on the work by Laure Pécry. And to start with, just a quick word about these particular structures. Otoliths are biocalcified structures that are present in the head of fish. And their role is to help the fish orientate themselves in its environment. In depth terms, they are considered as a product from the metabolism. And the definition of product in depth theory is that they are not considered structure because they don't require maintenance, but they're also not reserved because they're not readily available for growth or reproduction. Products in depth theory are the result of the contribution of three organizing fluxes, namely the assimilation, growth, and dissipation fluxes. For instance, feces receive a contribution from the assimilation flux, and respiration, or the release of carbon dioxide, is a weighted sum also of the three fluxes. And we assume that the creation of biocarbonates such as otolith is the result of two major fluxes, growth and dissipation only, not assimilation. And because we need to assess the contribution of these two fluxes, we introduce two parameters that will tell us what is the contribution of growth on one hand and dissipation on the other hand in the creation of this product. And so we can look at these otoliths in two ways. First, there is just their volume that we can track. And you have here an equation about the variation of volume, which is, as I said, a weighted sum of the contribution of these two fluxes, growth and dissipation. And we can also look at the opacity of this structure, and which is a very particular feature of these structure. As time goes and metabolism fluctuates, the opacity of the otolith will change. And the assumption that is made here is that growth, when there is growth in the organism, it will leave a, here in the image, a wider color, or a more opaque part of the otolith. But when no growth occurs, then we only have the dissipation contributing to its formation. And that's when you see darker marks where light can just go through. And so that's how we look at these otoliths in the organism. These structures are used often to age fish, and we will see a bit later on that it's not a without failure approach. And this work by Laure Pécry showed that. So back to the reconstruction problem, what we are trying to achieve here first is to build a model that a dead model that can help predict the growth of otolith, but also their opacity through time. And that's what they did using these two equations that I showed you. And from that, once this is built, we can invert the model and not use, for example, food as an input from the model, for the model, but use readings of opacity through time, or through the distance from the center of the otolith, to force the model in order to reconstruct the food availability, but also the growth of the organism. So here is a theoretical example to show you how this is done. So we are now in a construction mode. So the normal forward way of running a dev model, where we use the environmental conditions to force the model in order to predict length. So we have on the left here two theoretical variables for temperature and scaled food in the environment. And we can use that to predict length, but also to simulate the accretion of carbonates on the otolith. So it's the growth of the otolith, and also its opacity, and relate that to the data. And this is what this simulation can yield. You have on the right the simulated otolith, and a graph in the center for its in the lower center for its opacity through time, or more so through the distance from the center, but these distances can be then related to the time scale, as shown by this arrow that represent the periods where growth was not happening, basically due to lower temperatures and low food in the environment. And now, going backwards, we can use the otolith and their opacity to force the dam model and go back to the environment. And the method used to do so is based on the minimization of the squared deviation between observed and predicted volume of otolith and its opacity. So at each time step, you will have a certain value for the volume of the otolith and a certain value for its opacity. And the method used is a minimization method that best predicts what these volume and opacity will be at the next step. And what the length would be at the next step based on these values for volume and opacity. And so using, again, a theoretical example of temperature and opacity, we can reconstruct the length, which is exactly like the length predicted in the forward mode that we saw earlier, and also scaled food density. So there are been applications of this reconstruction method to look, for example, at different patterns observed in two populations of Atlantic cod, which show different types of different patterns in their otolith opacity, namely with a population from the Barents Sea showing translucent edge in the winter, and a population from the North Sea showing a translucent edge in the summer. And this work raised question about how we interpret the opacity variation in the otolith, and the use of this reconstruction approach allowed to determine that the patterns observed in the otoliths were not necessarily related only to a change in growth, but also to change in environmental conditions and migration patterns. And this raised question again about how we interpret these marks in the otoliths that are usually considered to be age mark, because the usual accepted explanation is that when there is no growth in the winter, this is where the dark mark is laid down in the otoliths. But if you are interested in this example, I encourage you to go check this study, which is very interesting. But that's an example, a very interesting example of how trajectory reconstruction can be used. All right, and for our last example, we will look at reconstruction of physiological rates and physiological state from shell growth data. And this is a work that is similar in its approach from the one we talked about before. For the otoliths, it doesn't use an otolith, it uses another calcified structure, which is the shell of a bivalve, here the great scallop. And what's interesting about this example is that, about this species, is that you can observe and you can measure the daily growth rate of these organisms by looking at the increments of their shell. This is a magnified, here on the left, it's a magnified picture of the growth increments of a scallop shell. And this is quite spectacular when you look at it, and it can be a very interesting to use this information as we are going to see. But it is a different process, though, than the approach on otoliths, because this reconstruction method is based on a rewriting of the depth growth equation into a linear form of equation. This work originates from an attempt to simplify how we deal with how we model growth in depth theory, in trying to reduce the number of parameters and being able to compare patterns in growth among organisms more easily. So it is a linear form of growth that only use 4 parameters that are listed here, and from that linearity in the growth equation, you can also retrieve the functional response that can be calculated from this scary equation here. But basically, the main interest of this approach is that first you reduce the number of parameters, and then you only need one type of data, here the length over time, to reconstruct the food availability. And as we saw earlier with spline functions, from which you can retrieve the first derivative, which is the growth rate from the length over time. Here we also going to use the second derivative, which is the acceleration. And because we can write now a depth model in a linear equation, we can rearrange different terms and retrieve some very interesting formula that, if you add only 2 parameters, allow you to reconstruct not only the functional response, but also physiological variables such as a state variable, the reserve, but also the reserve density, and some fluxes, some important fluxes in the depth model, which are the mobilization flux and the maintenance flux. So I will show you now an application of this method that we carried out along the Atlantic coast of Europe for this scallop species. And our main interest was there to look at data from remote habitats for which we don't have much information about the environmental conditions. Scallops can live from the north part of Norway to Portugal, and also on the continental shelf at depth superior to 200 meters. And in this area, sometimes it's really hard to retrieve data, but we sometimes retrieve some scallop shells, and we were interested to look at the environmental conditions from these locations. And you have here examples of growth trajectory. So these are daily shell growth rates. And in blue, you have a moving average, I think, and in red, a smooth version of this growth rate that is used to reconstruct food availability. And it's also this method has also been used to look at different forces for this bivalve, as I mentioned earlier in the example for the blue mussel. This reconstruction method can be used for that as well. And here you have an example of what these daily shell growth rate translate into in terms of functional response in different locations, going from Trena at in the northern part of Norway to Brest in Brittany, in France. And it's an interesting approach that can bring light on, can shed some light on patterns that are not necessarily easily available at first. And here we compare the mean reconstructed functional response along this latitudinal gradient and bathymetric gradient. And you see some interesting patterns showing that the life for a scallop at great depth is pretty similar in terms of food availability as what is the life for a scallop in northern Norway. You can also use this method to compare different age group, and this part of the study was mostly to validate the approach in showing that individuals of different age that are present in the same area and experience the same food condition would yield the same reconstructed functional response. And so not only can we reconstruct the functional response from this growth trajectory, but we can also reconstruct reserve density and mobilization flux and the somatic maintenance flux in these different locations. And this can also help us understand why we see different growth patterns in these areas. And for example, looking at the reserve density in the second row, we see that population both from the north of the distribution range and from the bottom of the Celtic Sea show a much higher reserve density than populations living on the coast in the southern side of their distribution range. And so there's a bunch of interpretation that can be made from these simulations and help us better understand the patterns that we see in the distribution of species and in their different growth patterns. And the last aspect of this study is to compare the reconstructed functional responses with proxies of food availability in the water. As I mentioned again, for the example with the blue mussel earlier on, bivalves feed on phytoplankton mostly, but they also filtrate other types of particles in the water. And this is a discussion topic actually that is quite present this year through the participants of the school. And this kind of approach can help better understand what food proxies are of relevance for a given species of bivalve, or what part of the year is most important for the growth of these bivalves, and what food is important at what part of the year mostly. All right, and to conclude, just a quick word on these different approaches. First of all, is that any of these approaches that I described rely on a set of assumptions. This is something that we deal often with models, but something that we need to be aware that these methods are applicable only if you are in certain conditions. The next thing is that, and I may have tripped through the words during this presentation, I apologize, but we here reconstruct mostly food ingestion and assimilation, not the actual food concentration in the environment or its availability. And this is so because the model is, when you invert the model, you can only go back to a certain point, and we are not able to model the selection process that may happen between what is available in the environment and what is actually ingested by the organism. Then you have sort of different, two different approaches within these different methods. Some of them are based on the minimization of deviation or error, and it's usually an easy and rapid method that can be used to reconstruct mostly functional response. Most of the studies available in the literature have shown that. But also some pretty detailed metabolic process, such as the work on otolith growth. Then you have approaches that are, shouldn't say equation-based, but because all of these are, but that really rely on a inversion of or rewriting of these the equations of the Deb model. Usually they require more and well-calibrated parameters. These are the first approaches that I mentioned in this presentation. Or in the last example about scallops, this rewriting of the Deb growth was based on the second version of Deb and not the last one, which was published in the 2010 book by Bascoiman. So all of them have advantages and inconvenience, if you wish. And the last point that you might have noticed too is that pretty much all the time during these in these approaches, we still need some environmental data, namely and temperature, unless you have some you are working on an organism that have a structure that can be used in isotopic analysis. Which is the case, for example, for scallops, in which you can retrieve the temperature from isotopic proxies, as is done for climate change research, when the scientists retrieve some isotopic signatures in different structure to recover different environmental conditions. And that's it for this lecture. If you have any question, we will have time during the live stations, but you can also email any questions you have, and I thank you for your attention.